So, dear colleagues and friends, for those of you who don't know me, my name is Tobias. I work in the clinic for rheumatology at the Charité, and also closely connected to the Deutsches Rheuma-Forschungszentrum. And I was asked to give an overview about B cells as a potential target in autoimmune diseases and provide some clinical data on the outcome of different B cell depleting strategies. And Henrik already alluded nicely why B cells are so important in the development of autoimmunity. And this is mostly also because they're right in the middle of this chronic feedback loop. And this is a nice overview from Thomas Dörner, summarizing the development of autoimmunity in lupus. And as you can see, B cells are right in the middle of this cascade. It's initially thought that signals from the innate immune system are amplified using soluble markers. And B cells then amplify this by being the source of autoreactive plasma cells that potentially then secrete autoantibodies. And these autoantibodies, together with antigens, form immune complexes, driving this vicious cycle. So the idea really is to have B cells and plasma cells in the focus of therapeutic approaches. And a short ABC of B cell function has been provided already. It's antibody production, antigen presentation, co-stimulation, cytokine production, and formation of ectopic lymphoid structures. And again, this is shown here. What is the role of B cells in their effector functions? They are involved in forming ectopic lymphoid structures, especially in Sjögren's syndromes, where you have ectopic lymphoid structures in the salivary glands. They are involved in antigen presentation. This is believed to be especially in MS, lupus, and RA. And they secrete inflammatory cytokines. In addition, also they may secrete cytokines involved in regulation, but they also increase inflammatory cytokines. And of course, they are the progenitor cells of the plasma cells that in the end secrete antibodies and form immune complexes. Now, what mechanisms do we have to prevent the development of autoreactive B cells? We all have autoreactive B cells, and our body gets rid of these autoreactive B cells by cell deletion and receptor editing. There are other mechanisms like intrinsic regulation and extrinsic regulation, and we can take use of those mechanisms for therapeutic interventions. For example, we can target CD20, 19, or 52 expressed on B cells. We could inhibit signaling cascades, and there's a list of soluble factors that can be blocked to target B cells. In former times, B cell targeting was mainly connected to conventional therapies. And I would like to show you some examples for the effect of conventional immunosuppression on B cells. But then the new era of biologic treatments emerged, and I would like to highlight the most important B cell depleting biologic therapies and new approaches, including plasma cell targeting. And here's a list of old immunosuppressive drugs, and for all drugs, there's a slight effect reported on B cells. And also on drugs that block TNF-alpha or IL-6 receptor. However, the effect is very mild. And as an example, here's a summary of the fate of B cells during treatment with cyclophosphamide, which is routinely used in the clinical routine for patients with severe organ manifestations. And what happens to B cells if patients receive cyclophosphamide monthly for 6 months. This is a work from the group of Thomas Derner. And as you can see, there's virtually no significant depletion of B cells, but what you can see is you have a depletion of plasma cells and a little bit on naive B cells, but not on memory B cells. And this is an important feature because we believe that memory B cells are those that are involved in autoreactivity and are the precursors of autoreactive plasma cells. So our treatment is not able to deplete autoreactive memory B cells. And this is the reason why we need to develop new drugs or concepts. Henrik already showed how B cells develop from hematopoietic stem cell during their development until the plasma cell. And you may have noticed that the expression of antigens on the surface of these B cells changes. One of the most important antigens expressed by B cells is CD20. They're expressed on B cells starting from the large B cell population until mature B cells. But as you've noticed, plasma cells do not express CD20. Nevertheless, CD20 is one of the most important drugs to deplete B cells, and it's a first in class, and it's been shown to be effective in various diseases, especially in rheumatoid arthritis, MS, ANCA-associated vasculitis, and lupus. And I would like to show antibodies that target CD20. The first-in-class antibody was rituximab, which is a chimeric antibody. The second-in-class antibody was ocrelizumab, which is humanized, and it's now approved for MS. Then you have a human ofatumumab antibody, and you have antibodies that target a type 2 CD20 receptor. And how do these antibodies deplete actually B cells? There are at least 3 different mechanisms by which anti-CD20 antibodies deplete B cells. The first is antibody-dependent cytotoxicity. Then you have a complement-mediated cytotoxicity and a direct cell death. And most importantly, the newer humanized anti-type 2 CD20 antibodies are more than 20-fold as effective for the ADCC, and also ocrelizumab is superior than the classical rituximab. Rituximab was initially approved for lymphoma, as a hematological disease, and over the years it has received approval for rheumatoid arthritis in 2006. Then it was approved as a first-line therapy for lymphoma. Recently it was approved for ANCA-associated vasculitis, and there is a new subcutaneous rituximab approved for lymphoma, and we wait for the approval also for our rheumatologic diseases for subcutaneous administration. The effect of rituximab was shown in a controlled randomized trial back in 2004, where people could show that methotrexate, which was the standard immunosuppressive drug, compared to rituximab alone, which is the light gray, or rituximab with cyclophosphamide, or rituximab and methotrexate, was less effective, or the other way around. Rituximab and methotrexate was significantly more effective clinically in patients with rheumatoid arthritis, and this is an ACR 50 response, meaning you have at least 50% improvement of symptoms. And coming back to the question, is rituximab able to deplete also autoreactive B cells? This group could show that you have indeed a reduction of rheumatoid factor, but only it's not complete, and of course you also have other autoantibodies that are secreted by plasma cells. Rituximab in lupus showed signs of efficacy, but it failed to show significance in non-renal and renal studies, but it shows in overall good data in lupus, and therefore it's recommended in patients who do not respond to conventional therapy by the EULAR and ACR recommendations as an established therapy. And as I've shown, rituximab is approved for vasculitis. This was based on this randomized controlled trial, showing that rituximab, in comparison to cyclophosphamide, is superior in clinical endpoints. And this graph shows you the B-cell depletion controlled as cyclophosphamide versus rituximab, where you can see that you have a slight reduction of B cells under cyclophosphamide, but it's of course more pronounced. It goes back to 0 under rituximab, and after a single regimen of rituximab, this B cell depletion remains for up to 180 days. What is the future for rituximab? We have biosimilars coming soon. This is shown here, and as you can see, it's more than one company that tries to build anti-CD20 antibodies. So this will, of course, make this drug cheaper in the future. So we switch to another antigen which may be targeted on B cells. This is CD22. It's expressed on mature naive B cells mostly, and it has a major impact on migration of B cells. It's adhesion molecule expression, and actually antibodies that target CD22 do not deplete such B cells, but rather they downregulate B cell activation and signaling. And it has been under investigation for several diseases. The group of Thomas Dörner was interested in looking on how the antibody targeting CD22 affects B cell signaling, and investigated Zinc phosphorylation. And if you pre-incubate B cells with anti-CD22, you see a decreased phosphorylation of Zinc in these B cells. And this is actually the rationale for using this drug in autoimmune diseases. Nevertheless, there was somewhat disappointing clinical data using this antibody as a therapeutic compound. Here is shown the EMBODY study, and this study compared placebo with nepatuzumab, which is the name of the anti-CD22 drug, with different dosages. And as you can see, the response rate is not much different between those three groups. So finally, this drug has no major clinical efficacy. There is some, but no major efficacy. The question is, can we use anti-CD20 treatment for all of our autoimmune diseases? And as shown by Hendrik, there are differences from disease to disease. One example is psoriasis arthritis, but there are other diseases that do not respond to anti-CD20 treatment. It's maybe a little bit small. So here's anti-CD20 treatment, and it's compared with other treatments such as anti-TNF or anti-IL-6, anti-IL-17, and this is shown for various diseases. Lupus, rheumatoid arthritis, vasculitis, and blue is positive results, and red is negative results. So in principle you have proven results of anti-CD20 in rheumatoid arthritis, vasculitis, relapsing remitting MS, and primary progressive MS, whereas you have no response in spondyloarthritis, including psoriatic arthritis and Crohn's disease. So there must be differences in the immunopathogenesis of those diseases. Instead of a direct targeting of B cells, one could use blocking agents that block survival factors that are needed by B cells and plasma cells. And I would like to show you two examples of blocking those soluble factors. One is a blockade of B cell activation factor, and one of blocking of APRIL. APRIL is believed to be a critical survival factor in plasma cells, as shown by Hendrik. It binds BCMA on plasma cells, and BAF is needed especially for a naive B cell survival. And the current state for drugs targeting either BAF or atacicept is that we have one approved drug for lupus, which is an anti-BAF treatment, but it shows no efficacy in RA. And what is the role of anti-BAF and APRIL targeting with TACI-IG? There are still trials in lupus. It shows no efficacy in RA, and it worsens inflammation in MS, interestingly. And just as a short reminder, survival factors such as BAF and APRIL bind three different receptors, which is TACI, BCMA, and the BAF receptor. And the BAF receptor is especially expressed on naive B cells, whereas BCMA and TACI are expressed on plasma cells. And the two drugs I mentioned, which is atacicept and belimumab, have a different way to target these soluble factors. Belimumab directly targets soluble BAF, so it's an anti-BAF antibody, whereas atacicept binds to the ligand, so it's a TACI immunoglobulin, TACI-IG. And this may explain the difference also in the biologic relevance, or because atacicept may block APRIL and BAF in the end, whereas belimumab only targets soluble BAF. What happens if you treat lupus patients with belimumab, so with anti-BAF? What is the effect on B cells and plasma cells? As expected, you have a decreased number of B cells in the circulation because BAF receptor is expressed on mostly B cells. And shown here is the median change from baseline. So you have a 60% decrease of B cells. And this is most pronounced in naive B cells. In contrast, memory B cells show even a relative increase in their frequencies. And this is because BAF receptor is expressed mostly on naive B cells. And then you have a slight decrease and reduction also of circulating plasma cells. But the most effect is shown here on B cells. And not surprisingly, you have only a mild decrease in immunoglobulins, which is shown here for immunoglobulin, and stronger for anti-DSDN antibodies. So for lupus, the current treatment algorithm includes belimumab, anti-BAF, if patients failed treatment with classical treatments such as MTX or azacitidine, while for organ-threatening lupus patients, belimumab is not approved. And here, as I said, rituximab is recommended, although the trials were not showed no significant improvement. So let's have a look at the data on atacicept in lupus. This is a trial reported here by David Eisenberg and David Wolfsey. They investigated atacicept in 2 different dosages and compared it with placebo. And this graph shows you the rate of relapses, because lupus is a disease that is characterized by flares. So they wanted to know, could you reduce the flare rate by this treatment? And you see there is a significant reduction of flare rates in the atacicept group with 150 milligram. And this is shown here. And as I said, APRIL is an important survival factor for plasma cells, and therefore the reduction of immunoglobulins is what we expect, and this is also shown. This is a mean absolute change IgG from baseline, and this is shown for IgG, IgM, and IgA, and for anti-DS10 antibodies. So you have a 50% reduction of autoantibodies and plasma cells and immunoglobulins. And this was something which was expected, but also which was associated with infections, because you need these antibodies also for infections. And this trial had to be stopped prematurely because there were severe infections that was leading to death. So at this time point, I think it's a very promising treatment, but this needs to be further investigated. We have a slide on B cells, and not surprisingly, B cells, median % change memory B cells for baseline, they are not depleted, whereas plasma cells, circulating plasma cells, are depleted. This is what you would expect. So in summary, blocking BAFF and APRIL, what have we learned? Anti-BAFF and APRIL in lupus shows bioactivity with reductions of B cells, plasma cells, Ig levels. Targeting B cell cytokines suggests distinct pathogenesis roles of memory B cells and plasma cells. Blocking BAFF is a promising target in lupus, and now it's approved, but it lacks evidence as a target in RA. And atacicept has a value in lupus, but needs further assessment and may be combined with immunoglobulin supplementation. But it shows no signs of efficacy in RA. So finally, I would like to come to plasma cell depletion. And one of the possible agents that can be used is proteasome inhibitors. Proteasomes are needed in the cells, and if they're blocked, you have cell cycle arrest, especially in those cells that secrete a lot of protein, and this is plasma cells. And therefore, it's not surprising that it's approved for multiple myeloma, so a disease where plasma cell malignancy. And then it has been tried to be translated to autoimmune diseases and with lupus. And based on preclinical models, we had bortezomib treatment performed as a clinical trial in our department. And it showed a very good clinical response. Here's just an example of skin manifestations under this treatment. And it also comes with a decrease in autoantibodies, as shown here for anti-DS10 antibodies. So really, plasma cells seem to be depleted. And in one case, we analyzed the plasma cells from the bone marrow and found that you have a 50% reduction of plasma cells in the bone marrow. On the other hand, the effect of B cells was very mild. So there was no difference in B cell numbers before and after this treatment. One of the drawbacks of this drug is that although you deplete plasma cells, they recur very quickly. So after the end of each cycle, we have a recurrence of circulating plasma cells. This is because the progenitors are not targeted by this treatment. What's the role of proteasome inhibition in RA? There was recently a publication showing that bortezomib improves RA in a patient that received stem cell transplantation later on for hematologic disease, and it shows good results also in preclinical models. So I think this is something which needs to be investigated. So what have we learned about plasma cell depletion with proteasome inhibition? It's clinically effective. It reduces autoantibodies and depletes plasma cells. But plasma cells regenerate very fast. So for those diseases, it needs to be combined with B-cell targeting therapies. The role of RA is unclear, but in general, I think it clearly demonstrates that it has a relevance in addition to B-cell depletion in autoimmune diseases. So we come to the overall summary. Based on successful trials with B-cell depleting therapies such as rituximab or belimumab, I think B-cell lineage cells are considered key players in autoimmunity, but the pathogenic role of the subsets differ between the entities. The contribution of reactivation of B cells varies versus continuous activity of resting autoimmunity, autoimmune cells by different compartments, which is memory B cells versus plasma cells, as well as B-regs, remain to be delineated. And the future may be a co-targeting of certain nodes of T and B cell interactions with additional innate immune targets and enhancing B-reg functions. And this is just a summary slide showing how we can target B cells in autoimmunity in their lineage development, and it summarizes again the role of targeting BAF in the early B cell checkpoint generation, anti-APRIL inhibition, and proteasome inhibition targeting plasma cells, and targeting B cells using anti-CD20. And this may be combined in the future also with drugs that target T cell activation or cells from the innate immune system to keep autoimmunity in check. So this was the final slide. I'm happy to take questions together with Hendrik. Thank you very much.
Yes. We do have time for discussion now, and as usual I will start since I have this possibility. So, as your last slide, what you said, and like in all B-cell development, you have several checkpoints where you actually can fail the tolerance. So, what is the role actually of chronic inflammation in breaking tolerance, and what is known about it? So, whether the chronic inflammation can break the tolerance, which could lead to pathogenic autoantibodies, or pathogenic autoantibodies always precede chronic inflammation.
Sounds like a classical hen and egg problem. I mean, I illustrated the direction of antibodies fueling the inflammatory reaction, but certainly since cytokines are also important factors in the differentiation, in the early differentiation of B cells, I could also imagine that inflammation impacts on the tolerance. I missed to say, but of course inflammation is a network effect of not only adaptive immunity but also of innate immunity. And so I think maybe it would take personalized approaches to determine what is going on and in which sequence it is going on in which patient, and maybe to deduct from this the fitting therapeutical intervention.
I think many things are unknown. For example, the role of memory B cells in the course of the disease. So we don't know do you have persistent memory B cell survival, and what's the role of these B cells? Do you have an activation of those memory B cells preceding flares of the disease? This is something which is unclear to my eyes. So, but I think it reflects that chronic inflammation causes, in terms of autoreactive B cells, also chronic autoimmunity. And if you deplete those B cells, you can arrest autoimmunity, but you may be aware that it may come back if you don't deplete the complete or the entire immunological B cell memory. Because also rituximab does not deplete every single B cell in your body. Any questions? Yes, please.
You mentioned the regular role of B cells, for example, in a model of multiple sclerosis in EAE. Do you know in other chronic inflammatory diseases, it is already known the role of regulatory role of these B cells? No?
There are additional investigations also outside EAE, but I can tell you now in detail. But I can provide like literature for further readings.
Do you know?
I think an interesting feature is what we've learned from clinical trials is that if you use anti-APRIL in MS, for example, that it gets worse. And we know that also plasmablasts may secrete regulatory B cells. So it's not clear if this is due to different components of BRACs, let's say in MS, in different compartments.
Do you know something about the immunogenicity of these drugs? Is this a problem, or?
You always have the chance to develop antibodies against your therapeutic antibodies, especially if you use chimeric antibodies. Therefore, the development goes into a development of humanized therapeutic antibodies, and this will be the future, I think. And of course, you have antibodies also detecting and targeting rituximab.
Yeah, as far as I know, there's also like the reaction of the immune system towards a therapeutic antibody is also a reason to switch the therapy, and either because of it causes directly a problem, or the drug becomes inefficient because it's basically the antibody is eaten up.
But is this checked? Like, do you check for the existence of antibodies, anti-drug in the patient?
In general, we don't check on a general basis because it does have no implication on your further therapy. So you would switch to therapy, but of course you could check for scientific reasons to address why develop one people antibodies and not the other and so on. But this is not performed on a regular basis.
Some further question?
You already, just a question. You already mentioned Rheuma. I didn't know that this is already an approved treatment for multiple sclerosis. This is in clinical trial, or is already approved?
Yeah. So maybe we can ask our colleagues from the neurology what the current. As far as I know, it's approved by the EMA, ocrelizumab in MS. But how is the state?
Yeah, it's actually, it's approved now, actually will come out this summer, and it seems that it's the most efficient drug in MS therapy whatsoever. So actually, even in which Tobias showed the primary progressive MS, it's effective even there.
It's expected to be on the market in fall somehow. So they postponed it, and it was expected first of all spring.
Okay, but so it would be the first possible main treatment for MS, progressive MS in this case?
Yeah, yeah. I mean, the effect is subtle, but it's there.
Okay.
So if there are no more questions, so you can always ask in private. The speakers are still here. And I would like to thank both of them for exciting talks and yeah, thanks a lot.
Thank you very much.